Hello everyone. Today I'm going to talk about the opportunities and perspective of the use of resistant varieties, which are one of the components to help viticulture meet the expectations for achieving new standards of sustainability. So let's get started with placing the current situation of resistant varieties in the right historical context. So there are hundreds of resistant varieties that have been produced over the past century in 4 areas of the world: North America, Europe, the former Soviet Union, and China, which have achieved market relevance and viticultural importance. So in North America and China, and in part of the Soviet Union, the interest in developing and using resistant varieties was mainly driven by the struggle of growing grapes in hospitable places where traditional wine grapes fail due to climatic extremes or high disease pressure. So there are a lot of examples of this in the Midwest of the United States, on the Atlantic coast as well, where historical varieties like Northern, Concord, or even Muscadine grapes are cultivated. In China as well, there are a lot of Labrusca and Amurensis hybrids that are directly used for cultivation. In Europe, the wines of resistant varieties needs to compete over the very same premium wine regions with wines obtained from traditional wine grapes. So resistant varieties do not have viticultural niches where they are the only one to withstand. So that's why grape breeding in Europe added a slightly different direction compared to breeding in other parts of the world. And the newest resistant varieties have in Europe high quality standards. So the trend that I have just described is also reflected in historical waves of breeding activities over time, which followed a sort of surge and pause pattern if we look at the number of released varieties per year. So the first 2 waves correspond to the efforts made first in the US to grow grapes with some vinifera blood in presence of the native pathogens, and then here in France to improve viniferas when the same pathogens arrived in Europe. So the third large wave from the end of World War II to the end of the century, which peaked around the 70s, corresponds to more and more cross generations to vinifera for restoring wine quality. So the productivity of grape breeding declined to nearly nil around the 90s as a natural tail end of this third phase, and then revived in the past 25 years, driven, in my opinion, by these 3 things: the need of bringing wine quality to excellence, the progress in understanding the genetics behind resistance, and the discovery of pathogen population dynamics. And I want to share with you in the next slide some data about these 3 points. So wine quality first. So if we focused on European resistant varieties, a massive effort has been done with improving wine quality, and a goal that from my personal point of view is nearly achieved. A few years ago, we compared white wines from 2 divergent clones of Sauvignon Blanc, one fruity, this one, and one herbaceous, and from a close relative of Sauvignon Blanc, Tokai Friulano, which is also known as Sauvignon As, and 4 resistant wines from resistant varieties, 2 offsprings of Sauvignon Blanc and 2 of Tokai Friulano. So this spider graph considers the rating in 7 attributes: finesse, intensity, structure, complexity, liking, familiarity, and willingness to buy. So none of the wines surpassed the fruity tropical Sauvignon Blanc clone, but the wine from one resistant offspring was very close to it. And the wines from other resistant varieties were similar in the panelist appreciation to the other Sauvignon Blanc clone, the dashed line, and to other vinifera wines. So this study also highlighted the fact that if we estimate fruit quality based on the area of the spider plot, which is represented here by these plots here, above a certain percentage of vinifera genome, there is not a linear relationship between the percent of vinifera genome itself in the variety and wine quality excellence. So this is exemplified by Sauvignon Ritos, which is this spider plot, which rivaled with Sauvignon Blanc for wine excellence, but is a bit more wild in genome composition than their peers, with approximately 83% of the vinifera genome. So a more recent study confirmed all of this for white wines. So the spider graph here of sensory analysis is from a group of wines from Vitis vinifera and a group of wines from disease-resistant varieties in green and in red. And the spider graphs are largely overlapping. So the conclusions of these authors was that the panelists were basically unable to distinguish between wines from resistant varieties and wines from the vinifera. So the same authors did something similar for red wines, finding a slightly different situation. In this specific case, wines from Cabernet Cortis, a resistant variety, were a little bit differentiated from wines from Cabernet Sauvignon, but this differentiation took also into account chemical differences in addition to sensory properties. So a more wider comparison is needed for drawing conclusion for quality among the reds. So anyway, these achievements in wine quality improvement were made possible by more and more backcrossing, which increases the vinifera background and reduces the parts of the genome that have residual DNA from the wild ancestors. So each resistant variety is somewhere at some point along this vertical path, which goes from an F1 hybrid, resulting from a cross between a vinifera and a wild grape, down to the most modern lines that have a nearly vinifera genome, which is supplemented with a few chromosomal segments that carry the resistant factors. So those red segments here that were originally donated by the wild relatives. So as we go down along this chart, the contribution of the wild is thinner and thinner. So this typically means that the wine quality increases, and the resistance is increasingly relying on fewer and fewer resistant factors. And this is true for disease resistance, but it also holds true in general for capacity to withstand challenging or new conditions, which are traits that are typically donated by wild ancestors and then get diluted along breeding generations. So nowadays, with the use of DNA sequencing, we can generate what we call introgression maps, which depict the 19 grapevine chromosomes in each variety, indicating exactly the species of origin of each segment, and if known tracts carrying known resistant factors are present or absent. So these funny charts illustrate this. So in the upper graphs, completely white portions indicate 2 chromosome copies of vinifera. Blue ticks in the right part of each diagram indicate 1 vinifera copy, like here, and 1 wild copy. And green ticks to the left part of each diagram would indicate 2 wild chromosome copies, which is not the case of any of these 3 varieties, which have at least 1 vinifera copy everywhere across the genome and 2 vinifera copies where they are free of blue ticks. So the lower part is a color palette which indicates the wild species of origin of the non-vinifera segments. So the gray is vinifera, green to blue, like this segment here, indicates an origin from an Asian species, and the type of green of blue indicates exactly which species. The red to deep pink plus cyanin indicates origin from North American species, and the violet red here, like this track, is from Muscadinia rotundifolia. So in the quest for wine quality, like in this case, the contribution from the wild is limited to a few regions around a few known resistant genes, and all the rest is vinifera. So in the quest for stronger resistance, like in popular and historical varieties in North America, these may be F1 hybrids like Catawba, in which there is one vinifera copy and one wild copy along all the chromosomes, or back crosses to wild grapes like Concord and Norton. So in these cases, no chromosome segment is a pure vinifera, and on the contrary, there are large chunks in the genome, like these, that are completely white, exactly those in which there are green ticks on the left part of the diagram and blue ticks on the other one. So by extending this analysis to a larger number of resistant varieties from around the world, it is clear that at this moment there is a full range of options that are available to wine growers, both for those who cannot compromise on the strength of resistance, who may use F1 hybrids like these, which have 50% of vinifera DNA and 50% of the wild, or even more wild genotypes, which have more wild DNA than vinifera DNA. Then those who place increasingly priority on excellence of wine quality can choose among lines with a nearly vinifera genome. And then there is the whole scale of intermediate options. So this brings me to the second part of my talk, which is understanding the genetics underlying resistance and exploiting the resistant genes. So basically breeding is now mainly focusing on 4 important diseases: downy and powdery mildew, black rot, which is caused by Guicnardia biduelli, and Pierce disease, which is caused by Xylella. So let's focus today only on downy. A long list of about 40 RPV genes and alleles are currently known. For some resistant genes like RPV3, multiple resistant alleles are known and identified as -1, -2, -3. So out of all of this, a few of them are exploited in a significant number of resistant varieties available on the market. RPV1, which is from Muscadinia rotundifolia, the allelic series of RPV3 from North American species, and then RPV10 and 12 from Asian species. So this means that there are large gene families of related varieties that share the same genes in different backgrounds. And the rest of RPV genes are mainly present in pre-breeding material, or they are being moved right now in more vinifera backgrounds, or they are minor resistant factors which are less significant for breeding. So in the past 24 years, grape breeders discovered that major genetic loci provide resistance to specific pathogen strain by producing protein receptors that convert recognition of the pathogen into programmed cell death and defense activation. So among all possible mechanisms illustrated in this slide, only one is predominantly responsible for resistance in nature, and exploited in breeding. So all major resistant genes are classic genes that provide recognition capabilities by matching pathogen-delivered effectors. And when there is this match, the plant that carries the proper receptor produced by the resistant genes is able to stop the pathogen growth. So multiple R genes provide multiple recognition capabilities, and this concept is at the base of the current process of resistant gene pyramidization. So breeders are working for bringing together in the same individual multiple recognition capabilities, which were previously present separately in different groups of resistant varieties. So the rationale behind is that if a resistant variety has two or more receptors, it will detect effectors produced by different pathogen strains, extending the protection against a wider range of pathogen strains. Or it will detect multiple effectors produced by a single strain, making the protection against that strain more redundant and more resilient to changes in pathogen effectors. So nonetheless, the pathogen populations are highly variable due to past mutation that may have accumulated over time, but they also evolve at the present time. So the avirulence genes in the pathogen accumulate random mutation in the population, and the effectors change gradually in amino acid composition up to a point that the effector changes so much in conformation that is no longer matched by the plant receptor. So this is a stepwise model of evolution in which a number of successive mutations add to one another before then they can have a significant biological effect. In addition to this, in the past few years, research has also shown that deletions of avirulence genes may occur to pathogen strain. So contrary to the previous case, this is a quantum leap evolution model in which a single mutational event may have a significant biological effect. So deleting that effector from that specific pathogen strain. So at the moment, there are a lot of questions open on this topic, whether such a kind of pathogen can do well without these effectors, if there is a price to pay for the pathogen in the long term of surviving without those effectors and so on. But from our point of view, this reinforces the concept that a fully informed and responsible use of resistant varieties is needed, involving all the actors along the production chain and keeping in mind these three essential rules for viner management: deploy resistant varieties, preserve resistant genes, and monitor pathogen populations. So far, breeding has exploited what is available in nature and what is available from natural evolution. Not all the genes and the alleles that we need to improve the crop species that we wish are available in nature or have evolved in the way that is useful to us. And here comes the possibility of modifying genes in a target way with the use of genome editing, which is part of a broader toolkit known as New Genomic Techniques, or NGTs. So genome editing is based on a sort of biological scissors, depicted here, which are delivered by an RNA guide to a precise location in the genome where they cut the DNA helix, exactly at that point. Then the break is repaired by natural repair mechanisms of the cells, but errors in the native DNA sequences are likely to be introduced. If this occurs within a gene sequence, the result is the inactivation of that gene. So the application of genome editing in disease resistance are typically for suppressing the production in the plant of proteins that are targeted by the pathogen for host cell entry. So the so-called inactivation of susceptibility genes. So the classical example is the inactivation of the gene MLO, which, if present, is used for powdery mildews to enter the host cell, including grapevine powdery mildew. And the second classical target of genome editing is something that is slightly different from susceptibility genes. It's a class of receptors, of repressors of basal defense. Here, the classical example is DMR6, which encodes an enzyme that is naturally present in grapevines, and it brings down salicylic acid levels in the plant. So the inactivation by genome editing of DMR6 increases the basal level of salicylic acid, and this enhances the activation of immune response, which should increase or reduce susceptibility, increase resistance or reduce susceptibility. So in general, the fact that sustainability in viticulture is right now at a critical juncture is witnessed by the fact that EU has just funded 2 large projects, which are called Grape Bread for IPM and Shield for Grape. The first one is coordinated by the French National Institute for Agricultural Research, l'INRA, and the other one is coordinated by the Italian National Research Council. Both of them are on grapevine breeding and the application of new genomic techniques. So this involves collectively more than 20 research and breeding institutes and companies in major wine producing countries in Europe, with an unprecedented concentrations of resources and expectations for innovative breeding program products in this specialty crop. These 2 projects have just started, and they are expected to use innovative tools, but in particular to bring conventional breeding and new genomic techniques together. So to conclude and to summarize, I think that conventional breeding is still central to grape improvement, because most of the traits that we want to improve, like adaptation to new climate patterns, resilience to extreme conditions, low input requirements, all have a complex physiology behind, and this can be only improved using the genome reshuffling brought about by sexual recombination. But biotechnology is a suitable complement for conventional breeding, so it will complement conventional breeding rather than replacing conventional breeding, especially by creating alleles that are unavailable in nature. And in particular, the sector of resistant breeding will particularly benefit from bringing together classical mechanisms of resistance, like the ones that are already in varieties present on the market, with new ones that can be made available by the use of biotechnology. So thank you very much for your attention.